All right, we're going to go over Chapter 8 and 9, which cover mitosis and meiosis. Both of these processes are about making new cells. So whenever you hear mitosis or meiosis, the goal is to make new cells. The main difference is, with mitosis, we're trying to make new body cells that are identical to the parent cell. So this is what we're doing when we're trying to repair a cut and we need new skin cells to replace the ones that were damaged, or just as we're having growth spurts and we're growing up, we need to make new cells, and they need to be identical to the cells around them. So that's the process of mitosis. Meiosis is a similar process. It's still about cell division, but in meiosis, we are making sperm and egg cells. In this case, the daughter cells that are a product of meiosis are not going to be identical to the parent cells. So mitosis, we're making body cells that are identical to the cells that are replicating. Meiosis, we're making sperm and egg cells, and they will not be identical to the parent cells. We're going to talk about mitosis first. That's Chapter 8. So when we're entering into this chapter, we have to make a distinction between the way prokaryotes divide and prokaryotic cells divide and eukaryotic cells divide. One of the first main things we'll notice is the difference in the way the DNA is represented, and that's because of the difference in the way it shows up. Prokaryotes have this circle of DNA, whereas we eukaryotes have chromosomes that are made up of really long strands of DNA. Our prokaryotic cell division takes place in 2 stages, and that's going to make up their very simple cell cycle. So the first stage for prokaryotes is to copy the DNA, which is called replication. That makes sense. When we're replicating something, we're copying it. So first they copy the DNA, and next they'll split into 2 to form daughter cells. And that process is called binary fission. So here we can see both of those things. In replication, we need to copy the DNA, and this is basically how that happens. This is a circle of DNA in a prokaryotic cell. There's going to be a point in that DNA called the origin of replication, where an enzyme can show up and unwind the DNA strands to open them up so that now this strand is available to pair with new nucleotides and make copies. And this strand is now available to pair with new nucleotides and make copies, so that we will have 2 complete sets of DNA at the end of replication. Next, we've copied the DNA, but we have to make our 2 separate cells. So that's when binary fission happens. This is when one cell divides into 2 cells, and they will both have those complete copies of DNA. You can see it happening here. Here's our DNA replicating. The cell will then elongate, it will start to cleave in the middle here, and that's whenever it will pull apart into 2 separate and identical daughter cells. The hereditary information in a prokaryote is stored in DNA, just like with us. So whatever information is being passed on to the daughter cells came from the parent cells. The prokaryotic chromosome is that single circle of DNA. They don't have our more complicated long strands that are condensed into those X shapes. And the DNA replication begins with that unzipping of the DNA, and it's at the point of the origin of replication. Origin meaning the start of replication. So that's the starting point for the copy that's being made. The new double helix is going to be formed by adding those complementary nucleotides to the exposed DNA strands that have been unzipped, basically opened up. And at the end of the process, the cell possesses 2 complete copies of the hereditary information. And then it can divide and we'll have 2 cells. Here you can see just a more zoomed in version of that happening. Here's our origin of replication. That's where our enzyme polymerase will show up and helicase. These enzymes are responsible for unzipping and allowing new nucleotides to be added. The new double helix is going to be formed by adding those nucleotides. So now we've opened up our original strand. You can see here's one part of our original strand and another part of our original strand of DNA, or circle of DNA. Once both sides are open, they're both available for new nucleotide pairs, and that's how we're going to end up with our two separate copies that are identical. Because you can see this nucleotide matches this nucleotide matches this one. They're going to have the same pairing at the end. After replication, the cell is going to grow, so it will elongate, so it can divide the replicated DNA molecules between those two cells. And whenever the cell reaches an appropriate size, that's when it starts to split into two halves. The new plasma membrane and cell wall are going to be added at a point between the partitioned DNA. And eventually the cell is going to then constrict into two and form those two daughter cells. Each of these daughter cells is going to be a complete living cell with its own DNA, and it will have the same amount of DNA as the parent cell. So they will be identical. They'll have all the same DNA and the same amount of it. Here you can see that happening again. We've got our single circle of DNA that is double-stranded. That origin of replication is going to allow for the beginning of our copy to be made. We're going to end up with two copies of DNA. Once we've got those two copies, the cell will elongate until this wall begins to form and the cell pinches in two, and then bam, we have two identical daughter cells. And that's when we've completed our binary fission. Eukaryotes are more complex in everything, and this is included. So our eukaryotic cells will contain more DNA than those prokaryotic cells, and the DNA, as we know, is packaged differently. So the DNA in eukaryotic cells is linear, and then we package it into really compact chromosomes. So the strands are extremely long, but they're wound so tight together and condensed into this X shape so that they're really easy to efficiently copy. And there is more than one chromosome in a eukaryotic cell, unlike prokaryotic cells that have just that one circle of DNA. Our eukaryotic cells will undergo two different mechanisms whenever our cells are dividing. So first we have mitosis. This is cell division that occurs in non-reproductive cells. So pretty much all the cells of our body that are not sperm or eggs. We call those body cells somatic cells. So that's important to remember. You do want to know that the non-sex cells in our body are somatic cells, and that's what we are copying whenever, or making new copies of whenever we're going through mitosis. In meiosis, we're still aiming towards cell division, but this is going to occur for sexual reproduction. These are called germ cells, like whenever you germinate a plant. So you could think of it that way when you're starting the lineage of, your offspring. So germ cells are what will end up producing our sperm and egg cells. We've got these stages of the cell cycle for this process of mitosis. First, we have interphase. Next comes prophase, then metaphase, anaphase, telophase, and cytokinesis. These are typically summarized so that people can remember them as PMAT. Cytokinesis is the final step where our cells will split in two. But when we're talking about mitosis, we're talking about PMAT. You can see here's our cell cycle. So interphase, the first phase that we listed, is where the majority of the time that our cell exists is going to be spent. During interphase, that's when our cells are going to just be performing their normal functions. They could be making their proteins, sending things out of the cell, taking things into the cell. It's when our cells are growing, and it's also when the copies of the DNA are made. So all of that happens in interphase. Mitosis begins with prophase. So you can see here, only 10% of the life cycle of a cell is spent in mitosis. And that makes sense because the majority of the time it's existing. But occasionally we do need to make copies of ourselves when we're trying to grow, when we're trying to heal. So 10% of the cell cycle, represented here in yellow, is spent in mitosis. The first stage of that is prophase, and then we go on through PMAT. Interphase is comprised, so this is our, let's go back. Interphase, the majority of our life cycle as a cell, is comprised of a resting phase where no cell growth is happening at all. This is the GO phase. So it's the part of interphase where nothing's happening. It's the hangout. We're not growing. We're not copying DNA. It's just a normal chill time where nothing is really taking place. That's the GO phase. There's also the G1 phase, and that's the primary growth phase. So whenever a cell has just divided and now it needs to grow and get up to size, that's going to happen in the G1 phase. Most cells spend the majority of their lifespan in this phase, growing. When we get to the S phase of interphase, that's when DNA replication occurs. So that's when the copies of DNA are made. And it happens right before mitosis begins. And then in the G2 phase, that's where we're continuing to prepare for cell division, and that also includes replication of the mitochondria and synthesis of microtubules that are going to be needed for mitosis. So that's all of interphase. That's where our cells are spending 90% of their life cycle. Now we're going to go into, once our cells are ready to divide, we either need more cells because we're growing, because we're healing something, or maybe we're replacing old damaged cells. Whenever we're about to go into mitosis, we're going into prophase. Prophase, you could think of P like prepare. You could also think pro like before, so before the other steps of mitosis, whatever helps you remember it. But in prophase, our chromosomes are condensing because we don't want to have a really messy pile of chromosomes when we're trying to make really clean, precise copies of them. So we need to condense our chromosomes so that they can easily be copied. And our nuclear envelope surrounding the nucleus is going to break down because we have to access that DNA so that we can make these copies of our cells. And also, that's when our spindle fibers are going to come from our centrioles in animal cells. You remember we talked about centrioles and their relationship in the cytoskeleton and how they are made of protein filaments that are able to contribute to the structure of the cytoskeleton and also work as a means for long distance intracellular transport when we're moving things across our cells. So our centrioles are making those microtubules, and those microtubules in this case, in mitosis, we refer to them as spindle fibers. So the centrioles will create these spindle fibers, and that's what's going to actually allow for the chromosomes to be pulled apart to opposite poles of the cell. Our next step is going to be metaphase. So you could think M for middle, because this is when all of the chromosomes are going to line up in the middle of the cell. In science, we typically call that the equatorial plane. So you could think about how the equator is the middle of the Earth, and the equatorial plane is the middle of the cell where our chromosomes are lining up. Once they're all lined up there, our spindle fibers that are coming from the centrioles will be able to attach and bind to the chromosomes. Then we can go on to our next step, anaphase. This is the A phase. You could also think of it as A for away, because this is when the centromeres will divide and spindle fibers will break down, starting at the poles on either side of the cell. And because those centrioles are breaking down, they're creating a pole that's pulling the chromatids in the middle of the cell apart to the opposite sides. Next, we have telophase. You could think T for two cells, because this is the time where you can really start to see when you're looking at the cells, like, okay, we've got two nuclei, and it looks like we're about to have two cells. We don't quite yet. There's still one cell, but it's getting bigger, and it looks like it's going to pull apart, and we've got two nuclei. So at this point, our chromosomes will uncoil, and we're starting to get a new nuclear envelope that is being formed around the nucleus. And it's also when the spindle fibers are starting to disappear because they already did their job. They already pulled the chromosomes to the opposite side of the cell, and they're no longer needed. But you'll notice at the end of telophase, we don't have two cells. We're just really close to that. And that's because the next part is cytokinesis. Cytokinesis is just the word for when the actual cytoplasm divides. So we've got all the copies in one cell here, but not quite two individual cells until cytokinesis happens, because that's the actual split of the cytoplasm that will end this process where we will now have two identical daughter cells. So here we can look at that in kind of the cycle as it's happening. Here's interphase. It's where our cells are spending the majority of their time. Our chromosomes are extended and in use during the G1, S, and G2 phases. You'll remember that the GO phase is where the cell is just hanging out. It's not growing, it's not making copies, it's not doing anything productive, it's just existing. Once the cell does need to make a copy, that will trigger mitosis. Mitosis begins with prophase, where the chromosomes will condense. So you can see we don't have a jumble of chromosomes, we have condensed chromosomes. The nuclear envelope that was present surrounding the nucleus is now breaking down, so those chromosomes can be more accessible. And here's our spindle fibers forming from the centrioles. Once that's complete, we go to metaphase. Remember, we're thinking M for middle, because our chromosomes are lining up in the middle of our cell, which we call the equatorial plane. After our chromosomes have aligned up on that equatorial plane, we can go to anaphase. We're thinking A phase, A for away, because these chromatids are being pulled to opposite sides of the cell. And then we go on to telophase. Sorry, people say telophase or telophase. You could say it either way. The chromosomes here are uncoiling, and we have a new nuclear envelope that's forming. And now you can see the spindle fibers are disappearing because they already did their job and pulled everything to the opposite sides of the cell. But it's not complete until we go through cytokinesis, where our actual cytoplasm is going to be cleaved in half, and then we'll have our two daughter cells. So when we're talking about chromosomes, cells that have two of each type of chromosome, meaning one chromosome from mom and one chromosome from dad, are called diploid cells. Di here is meaning two, so that's what it's representing, two sets of chromosomes from mom and dad. And that's basically all of the cells in our body except for sperm and egg cells. Chromosomes numbers will vary among organisms. So most eukaryotes will have between 10 and 50 chromosomes in their somatic cells, which is the body cells. Humans have 46 chromosomes. So in our diploid cells, meaning our regular body cells, our skin cells, eye cells, stomach cells, the cells of our nervous system, all the cells of our body except for sperm and eggs are diploid somatic cells that have 46 chromosomes. When we talk about homologs, also known as homologous chromosomes, they contain information about the same traits, but that information may vary. So let's make that a little more clear. Here we can see homologous chromosomes. It means that they're about the same size, about the same shape, their centromere is about the same location and in line with each other, and also they're coding for basically the same things, but those things might not be identical. So for instance, this homolog could be coding for hair type and this homolog is coding for hair type, but maybe this one is for blonde straight hair, maybe this one is for blonde curly hair. Maybe this one is for blonde straight hair, and maybe this one is for brown curly hair. Either way, the idea is these are very similar, but they're not identical. They're coding for the same type of thing, but not the same result. So that's what we're talking about when we're talking about homologous chromosomes. And then also I wanted to point out, when we're talking about a chromosome, an individual side to that chromosome, like this strand here, is called a chromatid. So each chromosome here has 2 chromatids. Over here, we've got 2 chromatids. One homologous pair. So we also need to talk about sister chromatids. And prior to cell division, each of those homologous chromosomes is going to replicate. And that's what's going to form 2 identical copies called sister chromatids. So let's go back for a second. Our homologous chromosomes, like these that are being copied, are going to be doubled. So the chromatid here has been doubled and the chromatid here has been doubled, which is what has resulted in our sister chromatids. These are called sister chromatids because they're identical copies of each other. So these strands of chromatid are both identical. These chromatids are identical with the exact same information. This chromatid and this chromatid are not sisters though, because you can tell they're coding for different things. Well, here it's just represented by the color of these, but they would be identical if they were sister chromatids. So that's an important distinction to make between homologs and sister chromatids. The sister chromatids are also joined together by a structure called the centromere. Let's go back one more time. Here's the centromere. So that's the point where the chromatids meet and join together. Centromere, you could think like center of the chromosome. And it's not always exactly in the center, but it is always the connecting point. So it could be down here, here, here. It could be pretty much at any point along the length of the chromatid. Humans have 23 pairs of homologous chromosomes, which means that we have 46 chromosomes in each cell. Because really, even though we have pairs like this, homologous chromosome, which is a pair of sister chromatids, we're only counting this as one chromosome. There are 2 sides to it, the 2 chromatids, but we're counting chromosomes by the centromere. So even though this is one chromosome, this is also one chromosome because we're counting by the centromere. So humans have 46 chromosomes in each cell, aside from the sperm and egg cells. And when each chromosome in the pair is replicated, this makes for a total of 92 chromatids. So even though we have doubled our number of chromatids, we're still saying we have 46 chromosomes because we're counting by the centromere. Also, this is interesting. We have karyotypes. So anytime you need really advanced medical testing for any reason, this is an option to give scientists more information and give your doctors more information about your genetic makeup and how that could be contributing to any type of ailment. There's the option for a karyotype, which is just an arrangement of all of the chromosomes in your cell, so in your body. And in this, chromosomes can be compared based on their size, shape, and centromere location. So we're comparing the different chromosomes. And the karyotype on the right is showing 23 pairs of human chromosomes. Here you can see these chromatids are not even attached, but they are still considered a pair. Our chromosomes are comprised of chromatin, which is just the complex of DNA strands and the protein that's associated with it to make that condensed chromosome shape. The DNA in a chromosome is one very long double-stranded fiber that extends unbroken for the length of the whole chromosome. So that's a really long strand of DNA. But to make it more accessible, the DNA is coiled around proteins in order to make it fit in a really small space, despite the fact that it's so long. Our DNA is going to be coiled around proteins that we call histones. The histones have the perfect positive charge to complement the negative charge that comes from that phosphate group of our DNA. And as a result, they have this affinity to each other. So the DNA will coil around a core of 8 histone proteins, and that's going to form a complex called a nucleosome. The nucleosomes, in turn, can also be coiled together to form an even more compact shape for the chromosome. You can see that happening here. So here's the way we tend to imagine DNA as its double helix strand. Here you can see it has the affinity for this histone protein that makes it wrap around it and coil up. The polarity between the DNA itself and the charge on the histone protein is what allows it to just keep coiling and wrapping up and then folding together more and more complexly into this chromatin loop. Eventually we end up with this scaffold protein shape, and in the condensed version, here are all of those nucleosomes made up of histone proteins and DNA that will be condensed into this chromosome shape. So, just to remind you guys, whenever DNA chromatin wraps around proteins called histones, that complex is called a nucleosome. In interphase, we are setting the stage for cell division. So we're not dividing the cell in interphase. But it is where our chromosomes are going to be duplicated. So that's where the copy of our DNA is going to happen. And even though it's not really visible to us, chromosomes do begin to wind up tightly in a process called condensation during interphase. The cell division that allows interphase is division of the nuclear contents, called mitosis. Sorry, I think I misspoke, but that the cell division that follows interphase is division of the nuclear contents. So basically, once we've copied our DNA, now we are going into mitosis, where our cell will be divided. Mitosis is a continuous process, but it is divided into these 4 steps that we already mentioned: PMAT, prophase, metaphase, anaphase, and telophase. A little more detail on prophase. In prophase, that's where we are condensing our chromosomes, where they can first become visible with a light microscope. It's also when the nuclear envelope is starting to disintegrate, because we have to be able to access that DNA. And it's when the centrosomes, which are made up of those centrioles, are going to start to assemble that network of proteins called the spindle fibers. Each cable in the spindle is made up of those microtubules. And some of the microtubules are going to attach to the chromosomes, which is going to allow for them to be pulled to opposite sides of the cell. Metaphase. We're thinking M for middle. The chromosomes attached to the microtubules of the spindle are aligned in the center of the cell. So all of our chromosomes are lined up on that equatorial plane, and the centromeres are aligned along that imaginary line that divides the cell in half. That's the equatorial plane. So all of our chromosomes are lining up in the middle on the equatorial plane in metaphase. Next we have anaphase. That's where our centromeres are splitting, so they're starting to break apart, and that's making a pulling motion from the opposite sides of the cell, kind of reeling the chromosomes apart, or the chromatids apart, to opposite sides of the cell. That's how our sister chromatids are being separated. In telophase, that's when our spindle fibers are dismantled, because they already did their job. It's also when the nuclear envelope is starting to form again, because we've got complete sets of chromosomes for the 2 separate cells. And then the chromosomes will begin to condense again, because we don't want to randomly leave some chromosomes out or have them too close to the middle. So that's why we are condensing our chromosomes within that nuclear area. And also, this is when the nucleolus starts to reappear, where you could see it under the microscope. I told you guys think T like 2, because even though we don't have 2 cells yet at this step, we can tell we're about to. You can see the 2 individual sides and the 2 individual nuclei at the end of telophase. Oh, sorry. We're going to go back to this slide. So here you can see a side-by-side of what's happening in the cell and what it looks like under a microscope, which is kind of cool. So here in interphase, this is where our DNA is replicating and beginning to condense, where the centrioles, if present, will also replicate and help the cell prepare for division. In prophase, the first step of mitosis, as you can see here, the nuclear envelope is breaking down so we can get to the chromosomes. The mitotic spindle is beginning to form, and it will be complete at the end of prophase. So you can see the mitotic spindles coming from our centrioles. Once those are completely formed, we'll enter metaphase. In metaphase, we're going to the middle of the cell. You can see here the chromosomes are lining up on that equatorial plane, and here as well. Also, the spindle fibers are attaching. They're not pulling yet, but they're attaching to those centromeres. Next, we go to anaphase. We're thinking A for away. You can see the chromatids being pulled to the opposite sides of the cell. So A for away. Chromatids are being pulled away to opposite sides of the cell. And also, this is when the centromeres are replicating, because even though we've got a chromatid going over here, there was only one centromere to start with. So we have to copy that centromere so that this chromatid is still complete and still a chromosome. Next, we go to telophase. This is where the nuclear envelope is starting to reappear. And the chromosomes are condensing. As telophase progresses, cytokinesis can begin. In cytokinesis, that's where we are actually cleaving those cells into 2 separate daughter cells, identical to their parent, and still diploid. So they still contain all of the genetic information that the previous cell that divided had. Whenever we do that split in cytokinesis, it occurs at the very end of mitosis. It's when we're cutting the cytoplasm in half, or roughly equal halves. And in animals, cytokinesis is actually occurring because of actin filaments that are contracting and pinching the cell in 2. So we can see that happening under a microscope when it happens, and we call it a cleavage furrow when we're talking about animal cells. In plants, it's slightly different, because instead of that cleavage furrow where you can see the pinched area where the cells are about to split, instead, in plants, you'll see a new cell wall is being laid to divide the 2 daughter cells. And we call that the cell plate. It grows at a right angle to the mitotic spindle, and it's right in the middle of the cell as well. So you can see those here. Here's an animal cell. You can see cytokinesis has begun. We've got our cleavage furrow. Those actin filaments are pinching the cell. They're going to keep doing so until they eventually sever this single cell into 2 separate cells. In a plant cell, like we see here, instead of a cleavage furrow, we can just observe the beginning of a new cell wall, which we call the cell plate, because it's not a full wall yet, but it is the beginning of one. So that's the cell plate that we would look for to indicate cytokinesis has begun in a plant cell. And our cells just don't live forever. So cells are programmed to undergo division about 50 times for humans. And whenever we're talking about cancer cells, really the main thing that defines them is uncontrolled cell division. That's the problem. It's just something got messed up in the DNA, there was a mutation, and as a result, there's no signal that is stopping division from happening. So we've got uncontrolled cell division. We just keep making cells that we don't need. That's how we end up with tumors. So when we're talking about controlling the cell cycle, it's controlled at 3 different checkpoints through the entire cell cycle. 2 of the checkpoints happen during interphase, which is that 90% of the cell's life cycle. And then there's 1 checkpoint that happens during mitosis. So the G1 checkpoint assesses cell growth to determine if a cell will divide or if the cell should just delay division, like it's not time to divide yet, or if the cell should enter the resting stage, the GO phase of interphase. The next checkpoint is the G2 checkpoint, and this assesses replication, which occurred during the S phase of interphase. Our third checkpoint is the M checkpoint, and this one happens during mitosis before continuing metaphase. So these are all checkpoints to decide, do we need more cells? Should we keep dividing? Is the DNA damaged? Should we just go into a resting phase instead of making more cells right now? That's what the goal of these checkpoints are. And you can see them represented here. So mitosis is just this blue arrow. So all of the copying of this cell and cleaving it in 2 is happening here. Cytokinesis, when the cell divides, is here. Now we've got a newly formed cell that is in interphase, which is the yellow, red, and green arrows in this cycle. Once we're in interphase, here's our G1 checkpoint, where we are assessing cell growth to determine if a cell will divide, if it shouldn't divide, if it should just enter the resting phase. And then you can see here, if it does enter the resting phase, that's when it goes into the GO phase. That is the resting stage, where the cell's not trying to do anything. But if the cell does need to make a copy, and that's the determination at this checkpoint, then the cycle is going to continue into the S phase. The S phase is the one where we can copy our DNA. After our copy of DNA has been made, the next checkpoint is going to be the G2 checkpoint. This is where we're making checks to that copied DNA. If there were problems with it, the cell might be signaled for destruction instead of mitosis if things go well. And then our next checkpoint is happening during mitosis, before continuing metaphase, and before the split of the cell in 2. So without any growth factors, cells are going to just stay in the GO phase. The thing that triggers cells to get out of that resting GO phase are those growth factors. Because in the GO phase, we're not growing, there's no replicating of chromosomes, there's no division happening. So in order to jump out of the GO phase, we need growth factors to trigger that. There are over 50 different proteins that do serve as growth factors, which can trigger the cell cycle and cell division. Platelet-derived growth factor, which is commonly called PDGF, is present in blood, and it triggers the neighboring cells to divide to heal a wound. So this is one of the most common growth factors that is talked about, PDGF. The cell proliferation signaling pathway is what binds a growth factor with a specific cell surface receptor. And once that binding happens, it will trigger an intracellular signaling pathway. And that's what's going to activate our proteins to start cell division. So just the simple fact that this growth factor landed on this cell surface receptor, on the surface of our phospholipid bilayer, is going to start a chain reaction that is going to allow a signal molecule to enter the nucleus and indicate that we should make a copy of this DNA and make another cell. So when we talk about chromosomes, there's an extra length of DNA on the ends of our chromosomes called telomeres. And these are created by the enzyme telomerase. So every time our cell divides, the chromosome is going to get a little bit shorter. And after about 50 cell cycles, the telomere will be gone, and that's what's going to signal the cell to enter senescence, which is cell death. So when we limit the number of cell divisions, we're actually lessening the risk of cancer, because the more times you copy your DNA, the more chance of error you have. And then if you continue to copy that error, the closer you get to a mutation, which could become cancerous. So that's why our cells don't just continue replicating endlessly. They go through about 50 cell cycles. Once the telomere at the end of the chromosomes are gone, that's when our cell is going to be signaled for cell death, senescence. And cancer is just basically a growth disorder of cells. It begins when apparently normal cells are starting to grow uncontrollably, and the result will be a growing cluster of cells called a tumor. Malignant tumors are invasive, and cells from malignant tumors can metastasize, which just basically means they leave and spread to different areas of the body to form new tumors. So here you can see metastasis. This is an example where we have our primary tumor that started in the lungs. So we've got lung cancer here. As a result of metastasis, the cancer cells have left the lungs and entered the bloodstream and traveled to other organs and started to proliferate there. So once you have reached metastasis, that's when your tumor is no longer contained to a specific area. It could have traveled throughout the body through the bloodstream. Here you can see what lung cancer cells look like at 300 times magnification. This is uncontrolled cell division. Cell division is regulated by proteins called growth factors. So those growth factors that trigger mitosis are also responsible for preventing cancer. Cancer can be caused by damage to genes that encode these growth factors. So if you damage the gene for a growth factor, you could change the way it does its job. This mutation causes damage to the genes, and the result is damage to the growth factor that should be controlling the cell cycle. These mutations that are cancerous can result from chemicals, viruses, and also environmental exposure to things like UV rays from the sun. There are two general classes of growth factor genes that are usually involved in cancer. We've got our proto-oncogenes, and these genes encode proteins that stimulate cell division. Whenever they're mutated, though, these genes become oncogenes, and then they can cause cells to divide excessively. So they're always proto-oncogenes are supposed to code for that cell division. But if they become mutated and become oncogenes, that's whenever there's no turnoff to that. So they're still doing their job of triggering cell division, but not when they're needed, just constantly. There's also tumor suppressor genes, and these are the genes that in normal circumstances will turn off cell division. So like, okay, that's enough cell division, we don't need any more right now in healthy cells. But whenever they're mutated, these genes could also allow uncontrolled cell division because they're not doing their job. They're not turning off cell division anymore. They've been mutated. Cancer results from damaged genes basically just failing to control cell division. Because the genes were mutated, they can't do their job anymore. In about 50% of cancers, the p53 gene is what has malfunctioned. And that could be because of chemical damage or radiation damage. But either way, the protein the gene encodes is not going to prevent tumor growth like it should because it mutated. Another really common cause of cancer is a mutated MDM2 gene, and this is one that becomes overactive, and its protein product is going to suppress p53. So even though this MDM2 gene isn't the actual one that's supposed to be handling cell division in this case, still, because it mutated, Now, its product is stopping the p53 gene from doing what it needs to do, which is prevent tumor growth. You can see that happening here. So here in our first step, this DNA has been damaged, could be from heat, radiation, chemicals. We've got our p53 protein. Cell division is stopping, and p53 will trigger enzymes to repair that damaged region. The p53 will trigger the destruction of cells that are damaged beyond repair. So it's like checking in to say, okay, no, this DNA doesn't look right. Let's go ahead and send this one to be recycled so we can use those parts for another cell. That's a normal p53 gene and how it should function. It's regulating cell division. It's making sure this is going the way it should. But if we have an abnormal p53 gene, then when our DNA damage is caused by heat, radiation, chemicals, and then we have an abnormal p53 gene, p53 protein resulting from that, then the p53 protein is going to fail to stop cell division. So it should have stopped this from happening, but it's damaged. It can't tell the difference between a normal cell and a cell with mutated DNA. So now the cell divides without repair, which means that the damaged DNA is going to be copied. And the damaged cells will continue to divide. If other damage accumulates, that's when the cell can turn cancerous. Now we're going to go over chapter 9, which is meiosis. So we just went over how we're making new cells to grow and to heal. Now we need to go over how to make new cells to reproduce. So we're going to be talking about how we make our sex cells, our egg and sperm cells. Again, as a refresher, both of these processes are about making new cells. The main difference is meiosis is when we're making sperm and egg cells for reproduction. So in this, our daughter cells will not be identical to parent cells, which makes sense because we all know that you can have the same parents as your sibling and end up looking completely different than them. There's all kinds of diversity, even when we're putting the same genetic pools together to create offspring. And that's because of meiosis and all of the variability that happens. So we've got our reproductive cells. These are our eggs and our sperm. Eggs for females, sperm for males. We call these gametes. So try to remember that word because it will be on the test. And our gametes, meaning our eggs and sperm cells, are cells that contain half the complement of chromosomes that are found in somatic cells, which is every other cell in your body. So our gametes, sperm and egg, are going to fuse together to form a zygote. The zygote is just what we call the moment fertilization has happened, when sperm has combined with egg. So now all of the chromosomes that was brought by the sperm and all of the chromosomes brought by the sperm and by the egg are both in a single cell. Now that's a zygote. You can see the zygote has 2 complete copies of each chromosome. The fusion of these gametes is called fertilization, and that's also referred to as syngamy sometimes. But if you could remember fertilization for this moment when we create a zygote, that would help you. The formation of our gametes has to involve a mechanism to halve the number, cut in half the number of chromosomes that are found in somatic cells. Because if we didn't do that, then the number of chromosomes would be doubling every time there was fertilization. If the sperm brought 46 chromosomes and the egg brought 46 chromosomes, then suddenly we're going to continue to double the amount of chromosomes we have. So we have to find a way to half this process, and that's what is happening in meiosis. It's a process of reduction division whenever we're forming our gametes, which basically means the number of chromosomes in the cell has been reduced by half, resulting in daughter cells with only one set of chromosomes. And this is going to ensure a consistent chromosome number throughout generations of a species. For us, it's 46. That's our magic chromosome number. So meiosis and fertilization are part of our sexual reproduction. Whenever we're talking about our somatic cells, they have two sets of chromosomes, from mom and from dad, and that's what makes them diploid. So that's the word for two sets of chromosomes, one from mom, one from dad. Gametes, the sperm and the egg, are called haploid because they have half the number of chromosomes of a diploid cell. Some organisms will reproduce by mitotic division, and they don't use gametes at all. That is called asexual reproduction. So there are kinds of reproduction where you don't need to introduce this genetic variability through meiosis. That is called asexual reproduction because there's no real genetic combination happening. An example of that is the binary fission that happens in prokaryotes. And other organisms are able to reproduce both sexually and asexually. So a good example of that is strawberry plants. They're able to go through sexual reproduction, meiosis, through their flowers. And they're also able to send out runners to start to grow and become larger plants through asexual reproduction. Binary fission, like I said, is an example of asexual reproduction. No genetic variability is happening. We're making clone cells. It's asexual. And here you can see what I'm talking about with the strawberry plant. So whenever it's reproducing by the flowers and there's variability that's introduced based on the pollen, that is sexual reproduction. But whenever this plant sends out a runner to start a whole new daughter plant, that is happening asexually. So there's no variability happening. This daughter plant is still a clone of the parent plant. And that's what makes it asexual. In sexual reproduction, our haploid cells or organisms alternate with diploid cells or organisms. So there are 3 types of sexual life cycles shown here. You can see this one with protists, most animals represented here, and most plants represented here. You can see all of them are cycling through diploid and haploid. Diploid is represented by 2N, meaning you have 2 copies of chromosomes from each parent. And then single N is showing haploid because you have half the amount of chromosomes as a complete individual of that species. That is also happening here with these animals. We are going through a cycle with all these diploid cells and still creating haploid gametes. Here you can see our plants after fertilization have created a diploid zygote and will have haploid cells and go through mitosis, asexual reproduction as well. So that was just an example to show you that all of these various organisms are going through these same processes. It's not just humans. In animals, the cells that are going to eventually undergo meiosis are reserved early on for reproduction. So these are in the ovaries of women and testes of men, and they're referred to as germline cells, which we mentioned at the beginning of this lecture. The germline cells are the cells that are able to produce eggs and sperm, so our gametes. Only the germline cells are going to be able to undergo meiosis to produce our haploid gametes. And here's the sexual life cycle in animals. So you can see the moment that a man and a woman create their egg and sperm, the moment that those egg and sperm meet in fertilization, where the chromosomes are brought from the father and from the mother to create a diploid zygote that has all the chromosomes needed to create a fully functioning human. That zygote will undergo mitosis so that the cell can divide and the organism can grow. Mitosis continues through childhood until the child has matured into a fully grown adult, with germline cells that can undergo meiosis and create either eggs or sperm that can then eventually enter fertilization and create a new zygote. So you can see 2N is when we're diploid, like when we're complete organisms that have all the genetic information. 1N is haploid, meaning you have half the information needed for an entire organism, like in the sperm, like in the egg. These gametes are haploid. When they combine together, that's whenever we've got a diploid zygote. I hope that the repetition is really helping this sink in. So I do want to make sure and be distinct between our homologs and sister chromatids, because you need to know which is which. Homologous chromosomes are basically a pair of chromosomes, one from each parent, that carry the same genes. So that could be eye color, could be hair type, could be body type, could have to do with your jawline, your hairline, whatever it is that gene is coding for. They're coding for basically the same thing, but it could be a different result of that hair type, of that eye type, of that jawline. So same but different. Our sister chromatids will be identical. So that's shown here. They're identical copies of a single chromosome that's created during DNA replication and interphase. And that's what makes them genetically identical to each other. So are homologs identical to each other? No. They're very similar, but they're not identical. Are sister chromatids identical to each other? Yes, they are. So, in summary here at the bottom, basically homologous chromosomes are similar but not identical. Sister chromatids are going to be exact copies. They will be identical. So the stages of meiosis. In meiosis, we've got meiosis 1 and meiosis 2. DNA is going to be replicated before meiosis starts, just like how DNA is replicated before mitosis starts. You have to make copies of the DNA before you can start making two separate cells. So that's going to happen before meiosis 1. In meiosis 1, that's when we're going to separate the homologous chromosomes. And in meiosis 2, that's when we're going to separate those sister chromatids. Whenever meiosis is complete, the result is that one diploid cell, this germline cell, has become 4 haploid cells. So these are all individual, so you could think of it as this is an egg, this is an egg, this is an egg, this is an egg. And they all have slightly different genetic information than each other and than the parent cell. And meiosis 1 is also divided into 4 stages, but because we have 2 divisions in meiosis instead of 1 division, let's go back. We've got 2 divisions. Here's our first division, 1 cell became 2. Here's our second division, 2 cells became 4. So that's what makes up the 2 different stages of meiosis 1 and meiosis 2. So meiosis is divided into meiosis 1 and meiosis 2. Meiosis 1 has 4 steps, all with a 1 following their phase name to indicate it's part of meiosis 1. When we look at the next area, the stages of mitosis 2 are the same way. Prophase in meiosis 2 is prophase 2. Metaphase in meiosis 2 is metaphase 2. Prophase in meiosis 1 is prophase 1. Metaphase in meiosis 1 is metaphase 1. So I just wanted to point that out as we go forward. Our first stage of meiosis 1, we've got prophase 1. This is where our nuclear envelope is breaking down and we're having homologs pair up and exchange segments. Don't worry too much about that because we're going to go into more detail. Metaphase 1 is when our paired homologs are going to line up on the equatorial plane of our cell. Anaphase, our homologs are going to be separated and pulled away to the opposite ends. In telophase 1, our chromosomes are going to gather up at each of the 2 poles of our cell. That's when cytokinesis is going to happen. Then our first cell will have divided into 2 daughter cells, and we will enter meiosis 2. So in meiosis 2, we will have prophase 2 first, which is where our new spindle fibers are forming to attach to those chromosome clusters. In metaphase 2, our individual chromosomes will line up in the middle along that equatorial plane. The spindle fibers are going to bind to both sides of the centromere from either side of the cell. In anaphase 2, those sister chromatids are pulled away to opposite poles. So the difference here is anaphase 1, we're pulling the homologous chromosomes away. So a chromosome with 2 chromatids is going to each side of the cell. Now in anaphase 2, we're pulling away the individual chromatids of that chromosome to opposite sides of the cell. And that's the moment where we have haploid cells. And then in telophase 2, the nuclear envelope will be reformed around each of the 4 sets of daughter chromosomes, so that cytokinesis can happen, and then we can end up with our 4 daughter cells. So now let's go into more detail on those steps. Prophase 1, in meiosis 1. This is where our homologous chromosomes are coming together as a pair, and crossing over occurs between non-sister chromatids of homologous chromosomes. So you see it says non-sister, so they're not identical chromatids, but they're going to cross over each other, quite literally. One side of a chromosome is going to lay over another side of a separate chromatid. And those chromatids are going to break in the same place and kind of trade sections of the chromosomes so that each chromosome will end up being more genetically diverse. The result is a hybrid chromosome. It's called a recombinant chromosome. And the pairing of that is held together by the cohesion between sister chromatids and the crossovers. You can see this is crossing over. So the visual really helps. It kind of just sounds like nonsense until you can see what's actually happening. But we've got our homologs. These are non-sister chromatids next to each other. They get close enough that they can overlap, and the cohesion that is going to kind of glue them together is what's going to allow for this exchange to take place, where suddenly now this chromosome has new genetic information and this chromosome has new genetic information. So now we've increased our genetic diversity by this step in prophase 1. Then we'll go on to metaphase 1. In metaphase 1, the orientation of those homologous chromosomes on the equatorial plane is completely random. So whether the chromosome is facing up or down or sideways, but each possible orientation of which way that chromosome is facing is going to affect the offspring cell, the daughter cell. It's going to affect which pole is going to result in gametes with different combinations of those parent chromosomes. And this part of the process in metaphase 1 is called independent assortment. So it really just means whatever orientation the chromosome is in before it's going to be pulled apart to opposite sides of the cell is going to determine another layer of genetic variability, because that changes what's going to be available in the next step. So here you can see that happening. Just the orientation of these chromosomes is going to determine what pairs are going to be left over at the end. Independent assortment is just another mechanism for increasing genetic variability. So you can see here we've got a paternal gamete and a maternal gamete. This is the diploid offspring. And then here are the potential gametes that could be produced because of independent assortment. So just basically, where were these chromosomes at the moment that they were pulled apart in anaphase 1? In metaphase 1. In anaphase 1, the chromosome pairs are going to separate and individual homologs are going to move away toward each pole. So our chromosome pairs are being pulled apart. In telophase 1, the chromosomes will gather at their respective poles to form 2 chromosome clusters. Cytokinesis will happen. That will divide our first cell into 2 cells, and then meiosis 2 is going to begin. So there will be a brief interphase after that, where there's no replication of DNA. It's just kind of a brief pause. And then meiosis 2 starts. And it's basically the same as a mitotic division. So it's basically, this part, meiosis 2, is basically the same process as mitosis, because we already got the genetic variability from crossing over and from independent assortment. Except the difference here is that the sister chromatids are not identical because of that crossing over. So it's the same functional process, but there is a difference because we don't have identical chromatids. So here we can see meiosis 1 happening, both in this representation and also what it looks like when we look under a microscope. So in prophase 1, our homologous chromosomes are going to condense and pair up. This is when crossing over occurs, where we get that exchange to end up with recombinant DNA. It's also when our spindle fibers are starting to form. In metaphase 1, our microtubule spindles are starting to attach to the chromosomes, and the homologous pairs that are lined up on the equatorial plane are going to be pulled to opposite sides of the cell. So you can see we're not pulling a single chromatid, we're pulling a whole homologous pair chromosome to opposite sides. And then they're both more diverse because of what happened in crossing over. In anaphase 1, homologous pairs of chromosomes are going to separate and move to opposite poles, so that's when they're actually pulled away. And then in telophase 1, we've got one set of duplicated homologous pairs at each side of the cell. And that's when the nuclear division begins and we go into cytokinesis for the first time. After that, we'll have that brief interphase, and then we'll enter prophase 2. In prophase 2, our chromosomes are condensing, recondensing, and our spindle fibers are starting to form again between those centrioles. Then we enter metaphase 2, where our microtubule spindle apparatus will attach to the centromeres of those chromosomes, and then the chromosomes will align along the spindle. In anaphase 2, that's when the sister chromatids are pulled to the opposite side of the cell. So not the whole chromosome, but the chromatid are pulled to the opposite sides of the cell. And then telophase 2 is when the chromatids arrive at each pole. So there they are, they've made it to their opposite ends, and cell division is going to begin. So once cytokinesis happens the second time, then we'll have made 4 haploid gamete cells. They all have half the number of the original diploid parent cell. So how meiosis differs from mitosis. First, meiosis has synapsis. So the homologous chromosome pair along their entire lengths and will be held together by cohesin proteins. That close association is what allows crossing over to happen. So that connection that they're making, we call synapsis. That point, let me go back. Well, I could do this slide. This point where this overlap is happening, that's synapsis, and that's what allows crossing over to happen. So that's definitely unique to meiosis, and it's providing genetic variability. Mitosis wants to end up with clones. Meiosis, we need genetic variability because that's what helps strengthen every species. So, synapsis, crossing over, is one thing that sets meiosis apart, and also the fact that it is a reduction division. So because meiosis involves those two nuclear divisions, but really only one replication of DNA, that's what results in the final amount of genetic material being passed on to the gametes being half that of the parent cell. So we are dividing cells, yes, but we are still reducing the amount of DNA. That's really all we mean with reduction division. Here you can see synapsis happening, where the sister chromatids of one duplicated chromosome are overlapping with another so that crossing over can occur. And then this is what we're talking about with reduction division. We have reduced the amount of genetic information in the daughter cell, but still divided. And we divided twice, resulting in 4 haploid gametes. This is a good overview if you want to pause, screenshot, take an in-depth look. I'm not going to spend more time going over this same stuff, but it's cool to look at it as a side-by-side when you're trying to compare. You can see the steps of meiosis 1, meiosis 2, and look how similar the process is to mitosis. You can look at the difference here where we see homologous chromosomes and the crossing over that takes place, which provides genetic variability. And you can see that meiosis has 2 cell divisions, while mitosis only has 1. Mitosis, we start with 1 diploid cell, end up with 2 diploid cells. Meiosis, we start with 1 diploid cell and end up with 4 haploid daughter cells. So our mechanisms of gene reassortment. There are basically 3 mechanisms of gene reassortment. And when we say that, we mean, how are we mixing up these genes to get more genetic variability? How are we getting that reassortment of the genes? The 3 main mechanisms for that is crossing over in prophase 1, independent assortment that's happening in metaphase 1, and then also a third point is just the randomness of fertilization. Whatever led you, or whatever led that individual to meet with the opposite sex at that exact specific time with that exact available egg and sperm, and they met at that exact moment and created a zygote, the randomness of that timing of fertilization is also a whole other aspect that's adding to genetic variability, because the sperm aren't all the same, the eggs aren't all the same, and now just the randomness of that moment has created even more diversity in the potential for offspring. So these 3 points are really good to remember when you need to think of what's providing that diversity in meiosis. Also, a reminder, independent assortment, when we're talking about that, we're just talking about the random orientation of the chromosomes as they line up on the equatorial plane in metaphase 1. So whatever orientation they're facing is going to affect which side they're pulled to, which other chromosomes are available within that haploid gamete, and that's also going to introduce more diversity. Sexual reproduction is conservative and also revolutionary. So we call it conservative because existing combinations of genes can be preserved because changes are not always naturally selected. So there are some genes that just continue to be passed on. They do not get phased out. They are conserved. It is also considered to be revolutionary because the pace of evolutionary change is going to be quickened by genetic recombination. We can make more changes faster and grow as a species and select genes that are more useful to our species specifically because of this genetic variability that's happening whenever we recombine these genes and provide more and more diversity to the offspring. So a really good example of gene-influenced natural selection, where a specific gene was passed on to offspring, is the melanin gene that is in peppered moths. And it's really an interesting example. So basically, before the Industrial Revolution, whenever there was such a big, huge push and new ability to really create things create products, create steel. Whenever we were entering the Industrial Revolution and starting to have a much higher incidence of pollution as a result of it, the peppered moths looked like this. And that's because they blend in so well on the bark of their chosen tree with that expression of that gene. But what we noticed after the Industrial Revolution, slowly, more and more of these peppered moths started to look like this. And we realized that it's because the gene that was being selected and passed on to offspring through natural selection was the mutation causing darker coloration. And that's because all of the soot from the industrial time that was covering the trees and making them look like this, even though they used to look like this, it meant that now the moths needed a better way to blend in to their new, changed environment. So the gene for darker melanin pigment was passed on because that helped them survive and blend in and avoid predators. So that's one example of this gene that was selected through natural selection that led to a shift in the population for the whole species. So that is all of chapter 8 and 9. Feel free to rewatch this, go back, pause it if you need, take notes. But really, the concepts that I went over are, you have to say so many things over and over again. Mitosis, meiosis 1, meiosis 2, all of the steps sound the same, and it can feel very jumbled. So I want to reassure you guys that if you watch these 3 videos, it will, I promise you, pull these concepts together in such a really fluid way. So first, watch this summary on mitosis. It's on Canvas under chapter 8 and 9. Next, watch the summary on meiosis. These are short, like 8-minute videos. And then after that, follow it up with this side by side, because it takes you through step by step in the perfect flow of what's going on in mitosis and how it's different from what's going on in meiosis. And it really will, I promise, help you lock these concepts in and remember what's happening at each stage and what the result is. If you have any questions, feel free to reach out to me. That's all for chapter 8 and 9.